my lab at Trinity College is interested in broadly how molecules inside cells work. And so the specific area that we're interested in is infection and immunity. So a cell is basically composed of various biomolecules like DNA, RNA, protein, lipids, etc. So proteins are really the workhorses of a cell. So they do everything from muscle contraction to moving cargo around in living cells. The particular area that my lab is interested in is infection and immunity. So when cells get invaded by viruses and bacteria, what typically happens is that the pathogens co-opt the cell machinery, including the proteins. And the way we go about understanding infection and immunity in living cells is we try to understand the structure of proteins, viral and bacterial proteins, and the human proteins that they target. So the methodology that we use is something called X-ray crystallography. So this is really at the frontier of biology and physics. You probably have all heard of medical imaging X-rays, and so it's a similar idea. We use X-rays, but they're much more focused, and they can give us information at the atomic level of what proteins look like. And so that's fundamentally our interest. So understanding how, for example, a viral protein is able to antagonize immunity can also provide ways for us to intervene therapeutically. And so, for example, a very good example of X-ray structure determination is the double helix of DNA. Understanding what the double helix of DNA looked like immediately provided insight into how heredity works. So one strand is the template for the other. Similarly, when we look at protein complexes, viral proteins, and their human targets, we can understand how they interact and possibly invent ways to modulate or to disrupt those interactions.